Hey GCSE biologists, in this video I'm going to be covering the entire of Module 3 for GCSE biology, which is infection and response. And just like last time, if you want a free set of flashcards on this topic, you can click the link in the description, fill in your details, and we'll send you them immediately for free. In the description, you'll also find a link to the GCSE Biology My Resources that have been written by myself and Joe Does Biology. These are full sets of study notes with full explainer videos, practice questions, and examiner tips and tricks. So go check those out too if you haven't done so yet. Okay, let's get going with the video. Let's go. So this is Module 3 for GCSE AQA Biology, which is infection and response. So this is all about communicable diseases. So first of all, what is a communicable disease? It's a disease that can be spread from one organism to another. So you could also describe it as an infectious disease or a contagious disease. These diseases are caused by pathogens. Now, the definition of a pathogen is a microorganism that causes disease. So microscopic living things that cause disease. There are 4 main types of pathogen that you need to know about. So let's go through them. The first type of pathogen is bacteria. Now, these are prokaryotic cells, which you should know means they don't have a nucleus. In fact, they don't have any membrane-bound organelles. So they don't have mitochondria either. They reproduce quickly using binary fission, which is a type of asexual reproduction. So when they reproduce, all of the bacteria will be genetically identical, or they will be clones. And they do that really, really quickly. It takes one bacterium about 20 minutes on average to divide and become 2. Now, how do they cause disease? Well, as I've just said, they reproduce rapidly in the body, but they release toxins or poisons that are going to damage your cells and tissues. The second type of pathogen is a virus. Now, viruses are actually not living things because they don't carry out respiration. And as we'll talk about in a minute, they can only replicate once they're inside a host cell. So they're not actually living things. They're also non-cellular. They are not made of cells. And this means they don't contain any common cell structures. So for example, they don't have a cell membrane. They don't have a cytoplasm. They are not cells. They're also much, much smaller than bacteria. So that's another comparison point that you can make. Now, as we said, they're not living things. And one of the reasons for this is they can only reproduce or replicate inside one of your host cells. And when they do this, they'll increase in number within the host cell, and then they will burst out of the host cell, which obviously damages the cell or destroys your body cells. Now, the third type of pathogen is fungi. Now, fungi, you don't learn much about their structure, but they are eukaryotic cells. They do have a nucleus. They can be single-celled or they can be multicellular, which just means they can consist of many cells. They produce spores, which they use for reproduction, and they grow on the surface of a host. Like, for example, they might grow and reproduce on your skin. The fourth type of pathogen is a protist. Now, protists are kind of a strange group of organisms, and they are eukaryotic, so their cells do have a nucleus. They're often parasites, which means they live on or inside another organism, and they depend on that organism for food and for survival. The example that you will need to know about for GCSE is the protist that causes malaria, which we'll talk about later. So they live in or on a host, and they may cause damage via toxins, so producing poisons, or they may damage your cells. So make sure you can name the 4 types of pathogen. Let's talk about some common ways that you can reduce the spread of a communicable or an infectious disease. And these are just kind of general methods to begin with, but we will be looking at more specific methods when we go through all the examples of diseases that are on your spec. So the first one is obviously really important, having good hygiene. So making sure you're washing your hands regularly with soap, specifically after using the toilet or before before preparing food. And I always find in exam questions, it is necessary to tell the examiner when you're going to wash your hands. Don't just say wash your hands. It's wash your hands after going to the toilet, before preparing food, and make sure you're using soap or some kind of antibacterial hand gel would be acceptable as well. Vaccinations. For some infectious diseases, you can be given a vaccination, for example, for measles or for tetanus, or you may have had your HPV vaccination. Isolating infected individuals. So if you have an infectious disease, isolating yourself, staying away from other people to prevent the spread of that disease from yourself to those other individuals. Destroying vectors. Now, this one is specific for malaria on your specification. Malaria is caused by a protist and it is spread by female mosquitoes. So if we can reduce the number of female mosquitoes or kill those mosquitoes, we can prevent the spread of that disease, or reduce the spread at least. Safe food and water practices. So making sure you're drinking clean water, decontaminating any water sources, hygienic food preparation, so using antibacterial sprays to wipe down any food preparation surfaces, washing your hands after handling raw chicken, for example. These would all be good ways to reduce the spread of infectious diseases. Let's go through now and look at specific diseases that are on your specification. So we're going to split them into viral, bacterial, fungal, and protist. So viral diseases are obviously caused by viruses. Remember, we've said these are non-living and non-cellular infectious agents that are much smaller than bacteria, and they are going to invade your cells, invade host cells in order to replicate, and then burst out of the cells, damaging or destroying the cell. So the first viral disease on your specification is measles. Now, some of the first symptoms of a measles infection would be a fever, so a high body temperature, higher than 37 degrees, which is normal body temperature, and also a red rash on the skin. Let's think about how it's transmitted or spread from person to person. Now, this is spread by respiratory droplets. So the virus itself is going to be present in the respiratory droplets of an infected person. And you can then catch that disease by inhaling these droplets from someone that sneezes or coughs near you if they're infected. So they're coughing, they're sneezing, right? In those droplets that they are producing, the virus will be present. And if you inhale that, you're inhaling the virus. So that is respiratory droplets. It can lead to serious complications, measles, such as brain damage, blindness, or even death. Now, you will have had a vaccination for measles. This is given to all young children in the UK, or as many as possible. And obviously, if you did have a measles infection, you would certainly isolate yourself from other individuals to prevent the spread to anybody else. The second viral disease, we're talking about HIV. Now, HIV is a virus. HIV stands for human immunodeficiency virus because it weakens your immune system, or it makes your immune system deficient. Now, the first symptoms of infection might be a flu-like illness shortly after you become infected with the virus. It is spread via sexual contact, sexual intercourse, or exchange of other bodily fluids. So, for example, via the blood, if individuals are sharing needles and one person was infected with HIV and someone else uses the same needle. So blood contact like that can also lead to the spread of HIV. Now, what are the effects or risks? Well, HIV virus actually replicates within your white blood cells, specifically within a type of white blood cell called a lymphocyte. Now, it destroys the white blood cells that it replicates within, and this leads to AIDS. It might take a few years to develop that condition, autoimmune deficiency disease, but it can lead to AIDS, which basically means a very much weakened or compromised immune system because it's replicating in your white blood cells. It's destroying your white blood cells. You have fewer white blood cells, so your immune system is severely compromised or damaged. And this obviously increases your susceptibility to other infections. So you may develop, for example, pneumonia, and your immune system cannot fight it because it is so weakened by that HIV virus. So how do we prevent the spread or control the spread? Well, obviously, because it's spread via sexual contact, sexual intercourse, using barrier methods of contraception like condoms is going to reduce the spread. And there are drugs or medications called antiretroviral drugs to prevent the development of AIDS. It actually slows down the reproduction of the HIV virus, but this is not a cure. It's just going to slow down the replication of the virus and hopefully prevent AIDS from developing. The third viral infection on your specification is actually a virus that affects plants. So let's just note that down. This is plant specific. So this is abbreviated to TMV, but TMV stands for tobacco mosaic virus. The reason it's called that is because it creates this kind of yellow mosaic pattern of discoloration on leaves. So the leaves kind of look patchy, they have yellow patches. Remember, yellowing of the leaves, there is a scientific term for yellowing of the leaves, which is chlorosis. So you should know that term. How is it spread? Well, it can be spread by contact, direct contact of an infected plant with a healthy plant. So the virus can spread that way, but it could also spread via contaminated tools that gardeners use or on the hands of the gardeners. It can be spread from plant to plant in that way. Now, what does it do? What are the effects? Well, this virus actually reduces chlorophyll production. And this is really important because it's come up on the exam many, many times. As it's replicating within the plant cells, it leads to a reduction in chlorophyll. Now, with less chlorophyll, there'll be less photosynthesis because you should know that chlorophyll is needed to absorb the light energy for photosynthesis. So with less chlorophyll, there's less photosynthesis. With less photosynthesis, that means there'll be less glucose produced by the plant. Now, you could go even further than that and say with less glucose, there's less glucose for respiration, which means there's less energy for the plant. There's less protein synthesis, which ultimately means the plant will experience poor growth, or you can call it stunted growth. Okay, now that's quite high level stuff, but it should make sense. If there's less glucose being made by the plant, less glucose for respiration, less energy, poor growth, but also with less glucose, they can make fewer amino acids because glucose is needed to make amino acids, right? With fewer amino acids, they make less proteins, and with less proteins, there's less growth. So ultimately, it's going to result in stunted growth. Now, how do you prevent the spread or control the spread? Well, if a plant has an infection with TMV, you should remove that plant. You could burn the plant to destroy the virus, and any equipment used during gardening or even your hands should be cleaned and sterilized before going to the next plant so that you're not transmitting that virus from the infected plant to the next plant. You can also keep your plants further away from each other. You can plant them further apart to reduce the spread, things like that. Let's talk about bacterial diseases. Now, these are caused by bacteria, which, remember, are cells, but they are prokaryotic cells without a nucleus. And bacteria cause disease by producing toxins that damage cells. So there are two on your specification. The first one is salmonella, which you may have had. Symptoms of salmonella: fever, so again, high temperature, abdominal cramps, vomiting, sickness, diarrhea. It's all those symptoms of food poisoning, right? How is it transmitted? Well, if you eat contaminated or infected food, and this bacterium is commonly found in chicken and in eggs, And obviously, if that food is contaminated or infected and it's not cooked properly, it's not cooked to a high enough temperature, then the salmonella bacteria will not be destroyed and you could end up with an infection. So you need to make sure that you're cooking your foods thoroughly and preparing your food in hygienic conditions. For example, sterilizing the surfaces in your kitchen, sterilizing the knife after you've been cutting raw chicken, and washing your hands as well after you've been preparing that food. Treatment, possibly antibiotics because it is a bacterial infection, but in most cases they're not needed. You would just be told to rest, drink plenty of fluids, and it might take you a few days to start to feel better. You might take some kind of painkiller type medication, drink lots of fluid to help with the diarrhea and the dehydration, but possibly antibiotics if it's a severe case or you've got a weakened immune system and you need the antibiotics to help kill the bacteria. Prevention and control. Now, this comes up a lot on the exam. So obviously hygienic food preparation. So you can give examples like wash your hands with soap before preparing the food and after preparing the food. Clean the food preparation surfaces with an antibacterial spray to kill the bacteria. Wash hands with soap after using the toilet, because otherwise, I know it sounds a little bit gross, but you could end up with feces on your hands with the salmonella bacteria present, and then you could go and touch surfaces or touch someone else or touch your mouth or whatever. So washing your hands with soap or using antibacterial hand gel after using the toilet is very, very important. Isolating yourself if you're infected to prevent the spread to someone else that is currently healthy. And interestingly, chickens are vaccinated in the UK to prevent contamination. The idea of this is if the chickens are not infected with salmonella, then the meat will not contain the salmonella. So then when we eat the meat, when we eat the chicken, the salmonella will not pass to us humans. And this idea has come up on the exam before, and many students struggle to make that link, right? But think about it with common sense. If the chickens are vaccinated, they're not infected with salmonella. The salmonella is not in the meat, so it's not passed to us when we consume the meat. Second bacterial infection is gonorrhea. Now, this is a sexually transmitted disease. The symptoms would be yellow or green discharge from the male penis or the female vagina, and also experiencing pain when urinating. It is spread via sexual contact with an infected person or sexual intercourse. Now, this would be treated with antibiotics. Obviously, it's a bacterial infection. Antibiotics kill bacteria. However, strains are becoming antibiotic resistant, which is making treatment harder. Remember, scientists are always working on developing new and different antibiotics to combat this antibiotic resistance, but it does take time to develop new antibiotics, and it is an expensive process. To reduce or prevent the spread, you need to use barrier methods of contraception, such as condoms, so the bacteria does not spread from an infected person to an uninfected person. Let's talk about our fungal diseases. So these diseases are obviously caused by fungi. Fungi thrive or reproduce rapidly in warm and moist conditions. So some of our prevention methods are going to be linked to trying to make sure the conditions are not warm and moist. The first one is athlete's foot, which can affect us humans, right? Symptoms would be itchy, red, flaky skin between the toes, cracked, peeling skin. I'm sure you've seen images, or maybe someone you know has had athlete's foot, right? And that can cause a burning, painful, stinging sensation on the skin. It can be transmitted through direct contact, so skin-to-skin contact of the skin of an infected person with a non-infected person. But it can be spread indirectly via surfaces, so using a towel that an infected person has previously used, or walking on the same shower floor, or walking on the floor in a public swimming baths where someone that's infected has previously been walking. Now, it can be treated with antifungal creams, or powders or sprays that will kill the fungus on your foot. But to prevent it, you can also try and keep your feet clean and dry, because remember, this fungus will thrive in moist conditions. So trying to keep your feet dry or using talcum powder to keep your feet dry could help. And wearing flip-flops if you're using a communal shower or you're walking around in a communal swimming baths, keeping your feet covered with your flip-flop. Not sharing towels, not sharing socks, not sharing shoes with anyone that might be infected would also be a method of prevention. Our second fungal disease, this is again a plant infection. So let's make sure we're clear on that. This is called rose black spot. It's caused by a fungus, and as the name suggests, black spot, it causes black or purpley spots on leaves. And then what starts to happen is the leaves will turn yellow around the edges and they will drop. The leaves will fall from the plant. And this is going to reduce plant growth because obviously fewer leaves, less photosynthesis. Now, this is transmitted via the wind, which may blow fungal spores from one infected plant to a healthy plant, or water, like rain splashes, which can wash the fungal spores from one infected plant to a healthy plant. It can also be spread via insects moving from plant to plant, or on gardeners' tools, or on gardeners' hands. Now, you can treat this with a fungicide. A fungicide kills a fungus. That's what fungicide means. But also you could remove any infected leaves. So when you see any purple or black spots appearing, you can remove those leaves, discard those leaves, or burn those leaves. Prevention and control of the spread. Obviously, as we've said, you'd remove and destroy any infected leaves or burn those infected leaves. You'd use your fungicides to kill the fungus. But another suggestion that you can give is avoid overhead watering, because if you're pouring water on your plant from the top, that water can act as a carrier. So the water will obviously trickle down, and if any leaves are infected at the top of the plant, the fungal spores could be carried by the water down and could then infect the leaves that were previously healthy further down the plant. So you want to avoid overhead watering so the water doesn't carry the fungal spores, and you just want to water the soil. You also want to avoid overcrowding plants. You want to plant them further away. So if it is windy and the wind is carrying the spores will not reach a healthy plant because the plants are more spread out, if that makes sense. The final group of diseases, these are our protist diseases. There is only one that you need to know about. Malaria is a disease caused by a protist. It's called the Plasmodium protist. Now, these protists are often found in areas with higher temperatures. They are not found in the UK, but in places like Africa, Asia, South and Central America, where it is warmer, there is high incidence of malaria. So let's look at this one. Malaria, as we said, is caused by a protist. The symptoms, recurrent episodes of fever, so lots of instances of raised high body temperature. In severe cases, it can be fatal if not treated. Now, let's just talk a little bit more about this. The protist, how it actually works, it invades liver cells, okay? And it multiplies or replicates within liver cells. Then these protists will leave the liver cells and infect red blood cells. Okay, so they're infecting liver cells, they're multiplying within those liver cells, then they're leaving the liver and entering the bloodstream where they're going to infect blood cells and again multiply or replicate, and then they burst the red blood cells. That's called cell lysis, but you don't need to know that term for GCSE. But that has been on the exam before, because think about it. If you're bursting red blood cells, you have fewer red blood cells to carry oxygen. And that links to the idea that less oxygen will then be delivered to your cells and tissues, which means less oxygen for aerobic respiration, which means less energy is released, which means extreme fatigue and tiredness. Okay. But obviously we will be posting a video very soon all about respiration, so we'll talk more about that then. But you should be able to make that link between the protist, the malarial protist, bursting red blood cells, lack of oxygen being carried in the blood, and therefore less oxygen for aerobic respiration. Right, how is it transmitted? This one is a little bit different. It's transmitted through female mosquitoes. Now, female mosquitoes act as vectors, which basically means they are carrying or transmitting the protist from one organism to another. When the mosquito bites a person, it will pass the malarial protist into their bloodstream, where obviously it will then go to the liver. But yeah, they'll feed on the blood from one person. They might pick up the protist, and then they'll feed on the blood of another person and inject the protist into their bloodstream. So they are carrying or spreading this protist. Be careful though. It's not the mosquito that causes malaria. The mosquito is just a vector, so it just carries the malarial protist from person to person. Now, in terms of treatments, scientists did develop the first malaria vaccine in 2021. So if you're visiting any countries where malaria is a problem, you can have the vaccine for malaria before you go. It can also be treated using an anti-malarial drug, but the protist is developing resistance to it. More important for you, or what comes up on your exam, is the prevention and control of the spread of malaria. Now, most of these are obviously linked to mosquitoes because it is spread via female mosquitoes. So we try to prevent mosquito breeding. Now, mosquitoes breed where there is standing or stagnant still water. So if we drain any areas where there is standing or stagnant water, we would be removing the breeding ground, which means less breeding of mosquitoes and hopefully fewer mosquitoes, especially fewer female mosquitoes. You could also use insecticides. These are chemicals that kill insects. So insecticides to kill the mosquitoes. You could also try to prevent the mosquitoes biting you. So using mosquito nets over windows, they can't get in. Putting mosquito nets around the bed so they can't bite you while you're sleeping. Wearing long sleeves, just generally keeping your body as covered as you can. Using insect repellents. Now, insect repellents are different to insecticides. Insecticides will kill the mosquitoes. Insect repellents will just repel them and hopefully stop them from biting you and maybe transmitting that protist. I'm just going to rub this out because at the bottom of this slide, we've also got a definition for you of a vector, just in case you could be asked for 1 mark. So a vector, and in this case we're talking about the mosquito, it's an organism that spreads disease from person to person or from organism to organism without getting the disease itself. So this vector would be the mosquito. It's the female mosquitoes only, and they are spreading the disease from person to person, but they are not affected by the disease themselves. Okay, so that's the term vector. Not the same as a vector in maths. I can't even remember what that is. Okay, let's move on now. Now we've covered the kind of infectious diseases that are on your spec, or the communicable diseases that are on your spec. Let's move on to how we try to prevent ourselves from getting ill from these contagious diseases. So first of all, we've got non-specific defense systems. These are the first lines of defense. They are non-specific because they will act against all pathogens. So they don't target specific microbes or specific pathogens. They will act in the same way for all pathogens, and the aim is just to stop the pathogens from entering your bloodstream, okay, just to prevent infection. So there are 4 main ones. There are more than 4, but these are the important ones. So the first defense system that you have, or defense mechanism, is your skin. Now, your skin is a physical barrier. It basically separates you from your environment. Yeah? Sounds obvious, but it is your physical barrier. It covers you, right? The aim of the skin, or one of the aims of the skin, is to prevent any pathogens from entering your body. And that's probably all you have to know for GCSE, but a little bit extra, it does produce antimicrobial secretions. So, for example, your sweat glands can produce enzymes to destroy bacteria. It also produces something called sebum. Your skin produces something called sebum, which does have a lower pH. So it helps to lower the pH and make your skin slightly acidic to prevent the chances that bacteria are going to be able to survive and thrive. I mean, you do still have bacteria living on your skin, right? We all know that. But because sebum is slightly acidic, it's a little bit more inhospitable to many of these harmful pathogens. So yeah, antimicrobial secretions, enzymes to destroy bacteria, sebum to make your skin slightly acidic. So hopefully harmful pathogens will not be surviving on your skin. But for GCSE, really, we can just say it's a physical barrier. It prevents pathogens from entering the body. Number 2, one that we all know about, in your airways. So I'm talking about your trachea, which is your windpipe, and then your 2 bronchi, one that goes into your right lung and one that goes into your left lung, your airways. They are lined with specialized cells. Now, there's some specialized cells called goblet cells that line your trachea and your bronchi, and they produce a mucus, which is sticky, so it traps pathogens. But you also have cilia. So there are cells that line your trachea and line your bronchi that are called ciliated, and I'm sure you've heard of these, ciliated epithelial cells. Remember, they have these cilia, these little hair-like structures on the top. And these hair-like structures, or cilia, move the mucus and the trapped pathogens up towards your throat to be swallowed or maybe coughed out. So the mucus and the ciliated cells are really working together, right? The mucus is sticky and traps the pathogens, and then the cilia will move and waft the mucus with the trapped pathogens upwards, away from your lungs, because you don't want a lung infection or chest infection, away from your lungs up towards your throat, and you'll cough it out or you'll swallow it. Now, swallowing it sounds a little bit gross, right? But actually it's fine, because your stomach is your 3rd defense mechanism. It contains hydrochloric acid, which is a strong acid. It's about pH 1.5 to pH 2. It is a strong acid, strong enough to kill most pathogens that you swallow in your food or in that mucus that's been moved up your trachea and you've swallowed it. This is an example of a chemical defense because we're using hydrochloric acid to kill the pathogens. Now, remember, your stomach is actually protected from the acid because your stomach has a layer of mucus inside to protect the muscular walls of the stomach so they do not get destroyed by this strong hydrochloric acid. Number 4, platelets. Now, platelets are fragments of cells. You've learned about them in Module 2 when you did the blood. They make up most of your... no, they don't. That's plasma. I was going to say they make up most of your blood. That's plasma. Ignore me. Platelets are fragments of cells. But what you need to know about them for this topic is if you cut yourself or you break your skin's barrier, these platelets will rush to the site where the cut is, where the damage is, and they will form blood clots. when the skin is damaged. Now, these blood clots will then dry to form a scab, and ultimately this is all about resealing the skin's barrier so the pathogens can't get in and they can't get into your bloodstream. You want to make that clot, form that dry scab, reseal the skin's barrier to prevent the direct entry of pathogens. Now, you might be thinking, what about nose hairs? What about eyelashes? I don't know. What about earwax? But these are not the important defense mechanisms. These are the ones that are important, these 4, and these are the ones that you need to learn about for GCSE. Now let's talk about the immune response. So what happens if all those defense mechanisms that we've just talked about fail and pathogens do get into your bloodstream? You're infected, right, with a bacterium or with a virus or with a protist. Now, the way this works, all cells, so your cells, as well as all pathogens, have protein markers on their surface. These are called antigens. Let's have a little look at a picture. So here is a pathogen. This could be, for example, a bacterium or a virus, but on the surface of the pathogen is an antigen. This is a protein. Yeah, so look at the definition. This is really important. This is getting harder now, right? So pathogens have proteins on their surface called antigens. Now, these antigens are specific. They will be a specific shape. And this is how your immune system can recognize which cells are your own cells and which cells may be pathogens or foreign cells. Because your body cells, let's imagine this is one of your body cells, they have antigens on their surface that are specific, they are a specific shape. And your immune system will recognize those antigens and will know they are your cells, they are self cells. But if a pathogen, let's draw a little bacterium here, the little flagellum, if a pathogen enters and makes its way into your bloodstream, the pathogen also has these antigens, these proteins on its cell surface, but can you see they're a different shape? They are specific to that pathogen and they are a different shape. So this will be recognized as a foreign cell because these are foreign antigens. They have a different shape to your antigens. So that's really important. That's how the immune system is going to recognize, oh, that's a pathogen, that's a foreign body, that should not be in my body. It's foreign, okay? The antigens are really important. So once a foreign antigen is detected, what is the immune system going to do? Well, the immune system's job is obviously to defend the body against pathogens that cause disease. If a pathogen enters the body, the immune system tries to identify it by those foreign antigens on the cell surface, and then it ultimately tries to destroy it, right? Now, your immune system consists of white blood cells. Really important. These are the cells that are going to recognize and destroy pathogens. So, how does it work? There are different types of white blood cell. The first type of white blood cell is called a phagocyte. Now, phagocytes are a type of white blood cell that will surround, engulf, and digest pathogens. So let me do you a little picture. So here is a phagocyte. Looks like a bit of a Pac-Man, but actually that's the cell membrane, that's the nucleus, that's the cytoplasm. Here's a pathogen which has got foreign antigens on its surface. Now, the phagocyte is going to recognize that those antigens are foreign and is going to engulf it. Do not say eat. I know my drawing looks a little bit like a Pac-Man eating a pathogen. Do not say eat. Say they engulf and digest the pathogen. Or you can say they engulf and destroy the pathogen like this. They kind of wrap their cell membrane around it. They enclose that pathogen. And then they use enzymes to destroy or digest it. Okay, so that's the first type of white blood cell. Now, the second type of white blood cell is called a lymphocyte. These are both white blood cells, okay? Just they do different things. So the second type of white blood cell is called a lymphocyte. Now, this is a type of white blood cell that produces antibodies. I'm going to have to rub my picture out there, aren't I? And these antibodies will be specific and complementary to the antigens on the pathogen. So I'm going to do another picture. So here's my pathogen. Doing my bacterium again, I'm doing the proteins on the surface, the antigens, remember they have a specific shape and they are recognized as foreign. Here's my lymphocyte. So my white blood cell, it's got a cell membrane, nucleus, cytoplasm. It's going to produce antibodies. Ooh. But not any old antibody. It has to produce the correct antibody, because the antibody has to be able to bind to that antigen. So this antibody, we can say it is specific for this pathogen. And a really nice word to use is it is complementary to the antigen. That just means it fits, it binds. A bit like how an enzyme's active site is complementary to the substrate because they fit together. Here we've got an antibody that is complementary in shape to the antigen, so it can bind to the antigen. Now, this is really important in exams, guys, because when you're talking about lymphocytes, you should be talking about them producing the correct antibody, the specific antibody, the antibody that can bind to that antigen. Okay, because you will need to use different lymphocytes and different antibodies for different pathogens because they will have different antigens on their surface. Okay, so once these antibodies have been produced, which by the way are also proteins, so antigens are proteins, antibodies are also proteins, they'll bind to the antigens and they will mark the pathogen for destruction. So then a phagocyte will come along and destroy the pathogen. At GCSE, you can literally just say, let me just phrase it how you guys can phrase it. So lymphocytes produce specific antibodies which destroy the pathogen. That's kind of a little bit of a lie because the antibodies don't directly destroy the pathogen. Really what happens is the antibody binds to the antigen and then it's a phagocyte that comes along and destroys the pathogen. But for GCSE, it's absolutely fine to just say lymphocytes produce specific antibodies and that destroys the pathogen. Okay. I just want to go back for 1 second, if you don't mind, just to, where was it? Oh, this diagram. I just want to make sure, because I know this is where students get super mixed up and I'm really trying to help you here. Just want to make sure you know the difference between an antigen on a pathogen and an antibody. So let's just write some definitions down. So an antigen is a protein on the surface of a pathogen. I mean, our cells do have them as well, but our antigens are recognized as self-antigens. You know, they're our antigens. But on a pathogen, an antigen is a protein on the surface of a pathogen that triggers an immune response, whereas an antibody is a protein produced by a lymphocyte, which remember is a type of white blood cell, which binds to an antigen to destroy the pathogen. The words are very similar, aren't they? Antigen, antibody, but the antigen is the protein on the surface of the pathogen. That's going to trigger your immune response. And then the antibody is produced by your white blood cells, your lymphocytes, and it's going to bind to the antigen to help destroy the pathogen. Okay, hopefully that is now nice and clear. we still have a little bit more to do here. We've talked about phagocytes. Phagocytes engulf and destroy pathogens. We've talked about lymphocytes. They make antibodies that are specific to destroy the pathogen. But lymphocytes can do something else. Lymphocytes can also make antitoxins. And all we need to say about this is antitoxins neutralize toxins. So remember from the start of our little lesson today, bacteria, we said they make us ill because they produce toxins that damage our cells. Well, lymphocytes can produce antitoxins which will bind to the toxin and neutralize the toxin, kind of cancel the toxin out so it will not damage our cells. Okay, so there's two things there for lymphocytes, the production of antibodies and the production of antitoxins. Let's talk about long-term immunity. So after you've had an infection, right, after your white blood cells have engulfed and destroyed or produced antibodies or antitoxins, some white blood cells can become memory cells. Now, memory cells give us long-term immunity, because memory cells are a type of white blood cell that will remain in your bloodstream for a long time. And if the same pathogen with the same foreign antigens enters your body again, memory cells can recognize the antigens. Oh, I've seen that antigen before. They recognize the foreign antigen and they can respond quickly. And the benefit of this is you will now produce large quantities of the correct antibody rapidly and for longer. Okay, so the memory cells recognize the antigen and they will be able to quickly make lots of antibodies to destroy the pathogen. It's all about the speed of the response. It's a much faster response. So this is why you often don't get ill twice from the same infection. So for example, you probably had chickenpox once. You created memory cells that recognized that viral antigen, because chickenpox is caused by a virus. And then when you get infected again later on in life, you can quickly make lots of the correct antibody to destroy that virus. This is also how vaccinations work. So they introduce the pathogen into your bloodstream so that you make memory cells. So if you get that infection for real later on, you can recognize the antigen. Let's have a look at this on a graph. So let's imagine the first time you are exposed to a pathogen, the first time you get chickenpox, right? The first time your immune system is exposed to the chickenpox virus with the antigen. Yes, you will start to make antibodies, but you can see from the graph, it's quite slow. There's a bit of a delay. Meanwhile, you're getting ill, you're showing symptoms, you've got your chickenpox spots. And then the concentration of antibodies does start to increase, but quite slowly. And you don't make that many. So the concentration of antibody does not get that high. Meanwhile, you are suffering from those symptoms, right? That would be your primary response or the first time you are infected with that specific pathogen. But then let's imagine many years later, you have a second exposure to that pathogen. So you get infected with the chickenpox virus again. You're exposed to that same foreign antigen. Have a look at the difference. Immediately, with no delay, you start to produce antibodies. The concentration of antibodies rises much more quickly, and the concentration gets much higher. That would be your second response, right? And this is because you've got memory cells that recognize the antigen and can quickly produce antibodies, and much more of them, much more rapidly. So hopefully you won't even know you're infected and you won't show any symptoms. You probably will not get the spots, the itchy chickenpox spots again. Let's talk about vaccinations next. So a vaccine is basically where they inject you with a small amount of dead or inactive pathogen. What we mustn't say, and I've seen this many times in student answers, we mustn't say they inject you with the disease, okay? They are not injecting you with the disease. They're not injecting you with a runny nose and a sore throat. They are injecting you with the pathogen that causes the disease. So they're injecting you with the bacterium or with the virus or with the protist. But so you don't actually get ill, the pathogen is dead or inactive, or you can say weakened. They might even just inject you with the antigens of the pathogen, the proteins from the surface. They might just inject you with those antigens because that's going to be enough to trigger an immune response, but you won't get ill. So these vaccinations won't cause disease, but because they're introducing the antigens into your bloodstream, those foreign antigens, then your white blood cells will recognize the foreign antigens, they will produce antibodies, and you will form memory cells to give you long-term immunity. Let's have a look at the steps involved in vaccination. So, how does it work? You are injected with dead or inactive or weakened pathogens. So they're introduced via a vaccine into your bloodstream. Your white blood cells are going to detect the foreign antigen and they are going to produce specific or the correct antibody. Remember, it's lymphocytes that produce antibodies. Lymphocytes are the type of white blood cell that produces antibodies. The antibodies are going to bind to the foreign antigens and destroy the pathogen. But some of these white blood cells will become memory cells, which is really important for long-term immunity, because if the same pathogen enters again and the same foreign antigen is recognized, memory cells can quickly produce the correct antibodies in large quantities and for longer. So the speed of response is much, much faster, and the concentration of antibodies in your bloodstream will rise much, much higher. So the pathogen will be destroyed before it can cause illness and before you experience, hopefully, any symptoms, or at least your symptoms will only be really mild. Okay, so let's move on. Let's think about evaluation of vaccination. So the pros, the benefits, and the potential cons or limitations. So advantages obviously massively outweigh any disadvantages. Vaccinations are used to prevent disease. So they protect individuals from really serious infections that could be life-threatening, such as measles, polio, or HPV. Now, HPV stands for human papilloma virus. You don't need to know that, but I'm guessing lots of you have had the HPV vaccination. This virus actually is linked to a higher risk of certain cancers, like cervical cancer in females. So this vaccination is usually given to children. I think it's when they're in year 8, to both boys and girls, to try and reduce the spread of this virus and hopefully reduce the risk of developing these kind of cancers later on in life. Polio, very serious life-threatening infection, but it has been eradicated or eliminated in the UK. There's been no cases of polio since, I think, the 1980s. And in 2003, the UK was declared free of this virus. So that's the benefit of vaccinations, right? You're not going to get these potentially life-threatening infectious diseases, and we are able to, in some cases, eradicate or eliminate the disease altogether from the population. Having vaccinations can also give herd immunity. This is basically where if enough people are vaccinated, so a large percentage of the population are vaccinated, it will reduce the spread of disease and even help to protect unvaccinated people, because so much of the population have been vaccinated. There's very few people that would ever be infected. So it's going to reduce the spread even to someone that is not vaccinated. As I said before, it can totally eradicate diseases. So some diseases like polio in the UK or smallpox have been wiped out completely.
And they are cost effective. I mean, it might cost a lot to carry out the vaccination program on a large scale, but that is cheaper than treating these serious diseases in the long term. Any limitations or disadvantages? Well, some people may have side effects, but they're usually mild. It might just be a little bit of swelling or a little bit of fever or nausea. Severe side effects are extremely, extremely rare. The injections themselves, you could say a small number of people might be scared of needles, they might be needle phobic, they might worry that they'll be painful, or you might get a small infection at the site of injection. Some vaccinations do need boosters because the effect of the vaccination wears off, and some years later you might need to have a booster injection. And finally, public resistance. So misinformation can cause people to avoid vaccines. And if fewer people get vaccinated, it does reduce their effectiveness. You might have heard of the controversy surrounding the MMR vaccination, which, this controversy was disproven. This vaccination was shown not to cause autism, but it did get into the public domain. It was in the media, and some people still have that in their heads and think that they're going to avoid the vaccination because of this potential risk, which has been disproven. Just a little bit more about herd immunity in case it comes up on your exam. As I said before, it's when a large percentage of the population is vaccinated. It means fewer people are infected, so there are fewer people to spread the pathogen. Now, this even helps people that are not vaccinated, whether that be through choice that they're not vaccinated, or maybe some people can't be vaccinated because they have other health conditions, they have weakened immune systems. Herd immunity can help to protect those people too, because with fewer infected people, it's going to reduce the spread of the pathogen to them. Okay, moving on to drugs and medications. Now, we're going to talk about two different types of drug. The first one is just a painkiller. So a painkiller would be something like paracetamol or ibuprofen, or, Calpol contains paracetamol. These do not destroy pathogens. They only relieve symptoms. So they might relieve your pain or your fever, your high temperature. So they make you feel better. They treat the symptoms, but they do not directly kill or destroy pathogens. They only reduce symptoms, but it's still your immune system and your white blood cells that are actually destroying the pathogen. On the other hand, we have antibiotics. Now, antibiotics are medicines that kill bacteria or stop them from replicating or reproducing. Remember, antibiotics do not work on viruses. They do not kill viruses. They only kill bacteria. That is really important. So any viral infections like influenza, which causes the flu, cannot be treated with an antibiotic. This is because viruses live inside your host cells. As we've said earlier, they replicate inside your cells. So the antibiotics cannot actually reach them without damaging your own body cells. So they're kind of hiding, right, within your host cells where they replicate. So the antibiotic cannot reach them. Remember, viruses are also not cells. So they don't have any of the cell components that an antibiotic would target anyway. Now, you should be able to name penicillin as an example of an antibiotic that kills bacteria. Penicillin was the first antibiotic discovered by Mr. Alexander Fleming. You should also know his name. And this antibiotic came from a fungus called the Penicillium mold. Why are antibiotics so important? One of the greatest things in all of science. Well, they have greatly reduced deaths from serious bacterial infections like pneumonia and tuberculosis or TB that many people did die from, but now with the use of antibiotics, they can be treated because the bacteria can be killed. They stop any bacterial infections from becoming life-threatening. So again, it's reduced the number of deaths dramatically from bacterial infections. And they are essential during surgeries where risk of bacterial infection is really high. Antibiotics are often given. And they're also essential for cancer patients that have weaker immune systems and could potentially get more bacterial infections. They can be easily treated with antibiotics. Let's talk about the problem, though, the misuse of antibiotics. Antibiotics have been over-prescribed by doctors in the past. They should not be prescribed for viral infections because antibiotics cannot kill viruses. They should not be prescribed for mild infections because they're not needed. Your own immune system and your own white blood cells can destroy those bacteria without the need for antibiotics. For example, with salmonella, often you do not need antibiotics to treat that infection. Also, not finishing a full course of antibiotics. Usually when you're prescribed antibiotics, you're told to take them, twice a day for 7 days. If you do not finish the full course of antibiotics, some bacteria may remain in your body even if you feel better, and they will start to replicate again. And the problem of all of these things is antibiotic resistance is developing. So bacteria are developing resistance to antibiotics. And what that means is the antibiotic will no longer be effective at killing the bacteria. There are resistant strains of bacteria like MRSA. I don't know if you've heard of that one. It stands for methicillin-resistant Staphylococcus aureus. But the reason I'm mentioning MRSA is because it's a bacteria that is very hard to treat, because this bacteria has evolved to develop resistance to many different antibiotics. Now, we're not going to cover the process of development of antibiotic resistance because that comes up in Module 6, which is a Paper 2 topic. So when you learn about Charles Darwin and evolution and the process of natural selection, you will use this as an example because antibiotic resistance has developed through natural selection, which is the basic idea that bacteria that are resistant will survive treatment with the antibiotic, they will reproduce or replicate, and they will pass on their genes for resistance. So over time, the population of resistant bacteria is increasing, which is a big problem because then they can't be killed by the antibiotic. But as I said, there's more of that to come in Module 6 when you cover natural selection in detail. Remember, scientists are working on developing new or different antibiotics to try and overcome the problem of antibiotic resistance, but this is very time consuming. It takes a long time to develop a new antibiotic and it is very expensive. One really important note from me, we should never be saying that bacteria become immune to antibiotics because bacteria don't have an immune system. They don't have white blood cells. They do not become immune. What we say instead is they become resistant to antibiotics. And that is actually through the process of mutation where the bacterial DNA changes at random and they develop resistance. And then they go on to survive, reproduce, and pass on that new gene for resistance. Okay. Something else that's on your spec then with regards to drugs or medicines, you should know that modern medicines, many of them were first discovered in nature from plants or microorganisms. There are some specific examples on your spec, so let's go through them. These could be some nice, easy 1-mark questions. So the drug digitalis, which is used to treat heart problems, that originally comes from a plant called the foxglove. Remember that. The next one, aspirin, which is a painkiller, so it relieves symptoms of pain, was originally from willow trees, specifically in the willow bark, but you can just say from willow trees. And the third one on your spec is penicillin, which remember was the first antibiotic discovered by Alexander Fleming. It kills bacteria. That one came from the Penicillium mold, which is a fungus. Now, not much more to say on this slide other than Remember the names of these 3 drugs and remember the name of the organism from which the drug originates. Final thing in this module is the story of drug development. So how do new drugs get discovered and developed, ready to be used by us humans? So new drugs and medications are tested for 3 key things before they can be approved and given a license and then sold in pharmacies or prescribed by doctors or used in hospitals. Those 3 things are, number 1, they have to be tested for toxicity. Basically, that means, is the drug safe? Is it going to cause any serious side effects? Is it toxic, right? So it's tested for toxicity. The second thing it's tested for is efficacy. Or put more simply, does the drug actually work? Does the drug do what it's supposed to do? So if the drug is used to treat migraines, does it actually help relieve migraines? If the drug is used to treat cancer, does it actually help to treat the cancer? Does it actually work? And the third thing is dosage. How much of the drug is needed? What is the optimum amount or the best amount of this drug to be given? Over what time period, what dose, how many times a day? Now, drugs are tested in 2 phases. We can split them into preclinical and clinical. So preclinical is like before testing on humans, and clinical is testing on humans. So let's talk about the first stage, which is preclinical, the very first stage of drug development. So this is where the drug is tested in laboratories. It will be tested on cell samples, on tissues, which are just groups of cells that work together to do a particular function. And they might be tested on live animals, usually small mammals, because humans are mammals. So small mammals like rats and mice should behave very similarly to these drugs. Now, the purpose of this stage is mainly to test for toxicity. So is the drug safe? Is it harmful? Is it dangerous? Are there any serious side effects? Now, it's really important that you can describe these stages and that you know what happens in each stage, what is the drug tested on, and what is the purpose of that stage. So preclinical, cells, tissues, live animals to test for toxicity or to see if there's any serious side effects. Then we move on to the clinical trials. So stage 2, the clinical trials will begin once the drug has been found to be non-toxic with no serious side effects. We'll start to test them on humans. Now, this is split into 3 stages. So stage 2, which is clinical trials, can be split into 3 further stages. Hopefully that makes sense. So on the first stage of the clinical trials, you're only going to use very low doses because we still really want to check for any side effects, right? So we're only going to use low doses of the drug, and we're only going to test on a small number of people. They're going to be volunteers that offer to be part of the drug trial, and they are going to be healthy. And again, this is mainly to test for side effects and toxicity. So is the drug safe? Are there any side effects? Are the side effects hopefully just minor by this point? The reason that we use small numbers of healthy volunteers, we can't test the drug initially on anyone that is sick. One reason for that is if they're already sick, this drug could make them worse if there are side effects. And also, if they're already sick, we might not actually be able to see the side effects because their other symptoms might mask the side effects. So we're just going to use a small number of healthy volunteers, so we're not putting them at any increased risk and we can see any side effects. The second stage of the clinical trials, so once we've now established the drug is not toxic, it is relatively safe, there are no serious side effects, we're going to start to test the drug on patients. Again, we're probably only going to use a small number in this stage, but we are going to test the drug on patients with the disease that the drug is designed to treat. Now, why do we need to do this? Well, at this stage, we're testing for efficacy. Does the drug actually work. So, for example, if this drug is used to treat heart disease, we're going to test it on individuals that have this type of heart disease to see if it actually improves their condition and if it actually works. And as always, we are still monitoring for any side effects, but hopefully we know by this point they're only going to be minor, so we're just monitoring for them. We just use a small dose of the medication to reduce the risk of any side effects. And obviously, we do need to use patience at this point because we are trying to see if the drug actually works, if it does what it says on the tin. And then finally, clinical trials phase 3. Now we need to start testing on large groups of patients of all different ages and ethnicities and genders, to check that the drug works on multiple people across multiple locations. So we're going to get lots and lots of data that is representative of the whole population. Now, at this point, we already should know the drug works. We should already have established whether there are any side effects. The drug is not toxic, but we can use this stage to determine the optimum dose, what is the best dosage to use, and carry on monitoring side effects just to check there are no rare side effects that might show up in certain individuals only. We can also check that the drug does not interact with any other medications, because some of the patients in this group, because it's such a large number of patients, might be taking other medication, and we need to know that the drug is okay to take with other medication, or should it be avoided if you're on that medication already. So we gradually increase the dose to find the best dosage. Ideally, we want the minimum dose that has the desired effect. So this is the amount you need to take, and it will work without any side effects. Okay? So remember, toxicity or side effects, efficacy, and optimum dosage are key, as well as checking, does the drug interact with any other medications? Now, once this has all been done, which can take many, many years, the drug will finally be peer-reviewed and approved. But let's just talk about that final stage. So you remember this stage here where we're using large groups of patients of all different ages, ethnicities across multiple locations. That stage is going to be done as a double-blind trial. Now, in a double-blind trial, some patients will get the real drug with the active ingredient, and some patients, half the patients, should get a placebo. Now, let's talk more about this. This is a double-blind trial. Double-blind means that the doctor that is giving out the drugs and overseeing the patients does not know who is getting the real drug and who is getting the placebo. And the patient also does not know whether they are getting the real drug or whether they are getting the placebo. That's why it's called double blind, right? It's a double blind trial because the patients don't know and the doctors that are overseeing it do not know. Obviously, someone has to know, right? The people behind the scenes that are looking at the data, they will know, so they can evaluate the effectiveness of the drug compared to the placebo. But we don't want the doctors that are dealing with the patients to know, because if they did know, they might influence the results. They might be biased because they really want the drug to work. And that bias might affect their reporting of the results and of their symptoms. So we're using a double-blind trial to avoid any bias, any influence on the results. And remember, half the patients are getting the placebo, but they don't know they're getting the placebo. The placebo has got to be something that looks like the real drug, so they don't know whether they're getting the real drug or the placebo. So if the real drug is a small white pill, the placebo would be a small white pill. If the real drug is an injection of what looks like a clear liquid, then the placebo would be an injection of what looks like a clear liquid, like saline solution or something. So it looks like the real drug, but it is not the real drug. It has no active ingredient. And using the placebo ensures that the results are not biased, because remember, the patients, the doctors don't know who has the placebo, who has the real drug. It also means that we negate the placebo effect. Now, the placebo effect is the idea that sometimes just the thought of taking medication can make you think you feel better. Have you heard of that before? Just thinking, oh, I'm having a drug, oh, I'm having a medication, this is going to help me. It can help you think that you feel better, and you might report that you feel better. But because half the patients are getting the real drug and half the patients are getting the placebo, only the patients taking the real drug should see real improvement in their symptoms. And that is going to provide proof or evidence that the real drug is effective. So we're really checking its efficacy. Does it really work? Do we see a real improvement in the symptoms of the patients getting the real drug compared to the patients getting the placebo? Okay. So it's really, really important. It helps to avoid bias and prove that the drug is actually working. After the drug has been tested, as I mentioned before, it does still need to be approved to be used. So the results of any drug trial and the conclusions from the drug trial will be peer reviewed. So this means that other scientists will look at the results and analyze the data. They might carry out their own tests, their own drug trial. And peer review is really important to check that all conclusions are valid and that the results or claims are not biased, because ultimately a drug company could stand to make money from the sale of a drug. So you want to make sure the claims are not biased and they are not just trying to make money, and that the drug is actually effective, it works, and there are no severe side effects, only mild side effects, if any. It is not toxic, has the right dosage, and does not interact with any other drugs. Guys, I hope you found this video useful.
I hope you enjoyed that video and, more importantly, found it useful for your revision. If you did, make sure to like the video and subscribe, because we're going to be posting more revision videos just like this very, very soon. If you do want to join one of the My Ed Space courses for GCSE biology, there is a link in the description that you can click to get more information. All of our courses have 2 live lessons every week with a workbook to go with the lesson. Every lesson has a homework and a video solution, and each month you also get a monthly assessment. So if you want to find out more about that, click the link in the description now. And remember, you can also click the link to access our free My Resources. Yes, totally free. We've got you covered for all your revision needs here at My Ed Space, and I'm looking forward to seeing you in the next video.